What we're going to do really quickly is just kind of give you a basic understanding of what's happening with the new therapies. And that's because we're going to hear a lot of discussion from the doctors about these new therapies. So to give you a little bit of framework, so when you go in, you're like, oh, okay, I kind of understand why they're talking about this and the difference that it is from what has been traditional therapy to date. All right? So the problem with traditional therapy or past therapies are really their chemotherapies. One, there's people that are still dying from their cancer. As Brian pointed out, there's a certain subset of patients that do not respond at all to traditional chemoimmunotherapy. And so that group of patients have definitely needed something else. Even people that do respond to chemoimmunotherapy, a number of those relapse. And there weren't necessarily good options for those people either, so there was lots of room in CLL to develop new medications. A few years ago when we were talking with Chris Fagan, one of the professors that actually will be speaking again at this conference, he talked about, 20 years ago when he was practicing for CLL, there was one therapy, chlorambucil, and so they would go to a conference for CLL specialists, and that's all they would talk about, is they would find a way to talk about chlorambucil any way that they could for 3 days. And now there's all kinds of different options, so it's really good that we're seeing these things come to light, and there's much better options, and we're keeping people alive longer. The other thing with the past therapies with chemo, it's really hard on the body. So if we looked at that age group. Still, the average age of diagnosis for CLL is in the 70s. People often have comorbidities, which means they have other diseases, other health issues that they're dealing with, which means that they cannot tolerate the chemotherapy the same way as younger people who don't have these issues. Okay, so if you have an existing heart problem, you can't necessarily have chemotherapy that's going to make that heart issue worse. Right? So doctors are always balancing between that do no harm and cure, right? So it's a very important balance. We can't give you a drug to treat your cancer if it's going to aggravate another condition and still cause a high probability of death or other more severe health issues for the patient. The toxicity, anybody who we all know chemotherapy, when we talk to patients or if you've experienced it yourself, has a whole wealth of issues associated with it. It's really unpleasant to go through. The nausea, the vomiting, the ongoing diarrhea for some people, your weakened immune systems. With FCR, some heart issues, etc. It's really hard to cope with. And so we needed something that was better, more tolerable for patients. And also drug resistance is a thing. So when we talk about people getting watch and wait, right, and starting with that therapy, it's because we know from previous clinical trials, if we treat people right away with FCR, which is the traditional chemotherapy, for instance, and they do go into remission, if their disease comes back, they cannot necessarily be treated with FCR again because they've developed a resistance to that medication. It doesn't work anymore. So when we had a limited number of therapies, once a patient relapsed, there was nothing else for them to do. So we would wait as long as possible to treat them so we could extend their life because we knew there was a high rate of relapse. Okay? And I'll explain a little bit more about what that means in a minute. So this is really the focus. It's like, okay, how can we improve the cure rate or the success rate without increasing the damage to the normal cells, right? How do we keep our patients alive with a good quality of life? So drug resistance, this is just really trying to help you understand. This actually has a flaw in the cell, but that's okay. In the slide, I'll figure it out and I'll show you. So at diagnosis, you may have one cell that is treatment resistant, all right? And after you've had your first treatment and you relapse again, there are more of these cells that are chemo resistant that have developed, all right? And by the third time, by the next time you get into, into refractory disease, basically most of your cells have become chemo resistant or treatment resistant. So it's just the process going through is like if you have one of these cells, this is going to be chemo resistant to begin with. What we do by getting rid of all the other cells is we allow this to multiply and grow. And it keeps doing it and keeps doing it. And you're not going to be able to handle that treatment anymore. And we have to find something new to treat a patient with. Does that help make sense?
And do you refer to all chemos as alike? Or, for example, first treatment like FCR and second treatment BR, would it be different?
Not necessarily. Even with some of the newer therapies, we're still seeing treatment resistance develop, right? So it can be with lots of different therapies. We saw it more, at least we saw it more because we have longer term data with the traditional chemotherapies that we've been using. We're starting to see it with some of the new therapies, but the data is still short. So we have to wait and see what that is looking like long term to actually figure out what that's going to look like from a treatment paradigm for a patient to see where we should treat and what we should treat with.
Go on. Sorry, Brian.
Yeah. So what's happening here in the chemoimmunotherapy is that this is why we say you have to test before each treatment, because in the first set you may have measured negative for 17p. It wasn't meaning that 17p wasn't there. It was just at such a low level that our tests couldn't detect it. But when you wipe out the competition, when you spray for the cockroaches, and the cockroaches come back are the ones that's resistance for what you sprayed for. Now, when you measure, you find 17p, and maybe it's in 20 or 30%. And then another time you measure it, it's 90% of the cells. And none of those cells are going to respond to any chemo, because 17p, if you're 17p, you don't respond to bendamustine, you don't respond to fludarabine, you don't respond to chlorambucil, you don't respond to any chemo. We're also finding that there are specific mutations that confer resistance to the new drugs, like ibrutinib and venetoclax. And those tend to develop over time too. And it's a different pattern and there are different things, but we're finding those are developing too, so that's an issue. So one of the theories, and this is another big fancy word called is that we should treat this orthogonally. Orthogonally means at right angles to each other. We hit the cancer with two different types of therapies. So one says, I don't care if you're resistant to ibrutinib, I'm going to work because I work on an entirely different pathway. I don't care if you're resistant to venetoclax, I'm ibrutinib and I work in an entirely different pathway, putting those two together. Or I'm venetoclax and obinutuzumab. So we're using two different drugs that work entirely different ways to overcome resistance. That's what's starting to happen.
Yeah, and to see, so if we can get rid of everything in these early phases, it stops this happening, right?
Can I just make one point just from when we started out, Jan, in 2011, and we were trying to find treatment options for what we noticed from, again, the time gone past when we look back on the forums, that a lot of the patient community that were discussing this with each other were saying about chemotherapy, 3 strikes and you're out. And that's what that slide was, that's just what it reminded me of. You have chemotherapy once, you might get a remission for a few years. You have chemotherapy the second time, you won't have a long remission, and the third time it won't work. And that's generally the way, and that's why these novel therapies, new treatments are so important. You know.
I'm going to talk about chemotherapy. We've been using chemotherapy for the last, 30, 50 years. And it's, I'm sure by now, something, we do something else apart, apart of this. Because sometimes it's not the disease you are dying from. You are dying of the, all the chemo that you
Yes.
get into your body.
Yeah.
So, after 30, 50 years, it's, sure, with all the money it's been invested in, finding out another kind of treatment. I don't know, it's, I find it
Frustrating.
frustrating, exactly.
Yes.
Exactly.
And we will say, CLL has only become a disease that has had a lot of research in the last few years. It was kind of ignored for a while. And it doesn't mean that there wasn't some research happening. It just wasn't as much research. And so when we saw more effort put into it was really when we started to find these new targeted therapies. And we'll look at a second at kind of a map of a B cell and how they figured out what targets that they were going to use. But it is fairly recent, and we admit. But it wasn't like there wasn't work happening. It just was like they were struggling with finding the right drugs that were going to work within CLL.
I think actually just to say the reason that CLL in research, and I've heard this at a few conferences from the doctors, and they might say it, is that a CLL patient at any given time when they come into the hospital and they give their blood, there's live cancer cells in the lab that they can work on. And you have an endless supply of these cancer cells that can keep, where if you have a patient that has a solid tumour, you have that solid tumour once to work on and then it's gone. And I think that's the reason. And CLL has actually become very, very important to all of the other research into other cancers and all the targets.
Blood cancers in general.
Blood
yes.
cancer in general, yeah.
Yeah, but you know, even with that, if we're looking at when, I'm going to name names, but Dr. John Bird, who is a leader in the CLL research community, when ibrutinib came to light, right, when it was first put out into the CLL world, he was convinced actually it was going to be curative, which is interesting because even those key researchers, when discoveries are made and they're looking at that initial data that's coming out, they're like, this is it, this is going to cure this cancer. And then it didn't. It's life extending, and that's good, but now that it's been in the world longer, we're seeing the relapses happen for some people, and other people are still doing fine on the medication, but it's giving them a new way then that they have to focus their research to account for what they're seeing. And we don't get that until we have it over time and within a bigger community than we're seeing within a clinical trial. Arvand, I know you've been waving and waving, so.
I want to share one good news. 1 week ago, FDA approved venetoclax plus Imbruvica.
Yeah.
After 1 year, 95% molecular remission. It's fantastic result for CLL. And maybe soon we couldn't speak about this slide.
Yes, maybe, but it's 1 year. Yes.
On molecular, we can speak about molecular remission in CLL.
Yes, and there's lots of cool things happening when you talk to patients, and you'll hear when the doctors are talking. One of the things they'll talk about is if there's still a place for traditional chemotherapy in modern day treatment. So that's why this is coming into play, is because, as Brian said, for some people, traditional chemotherapy is giving them a 14-year plus remission. So it's a limited time treatment compared to a lot of the new novel therapies, which are an ongoing therapy. So is that the best place? So we're going to hear the doctors talk about this.
You must remember also that the cost of chemotherapy is very, very low, while the cost of the other therapies is very, very high. So sometimes it's important to still have it for those populations where you cannot give them the expensive treatments.
As long as you know it may work for them. So when Brian is talking about the testing has to happen, because no matter how cheap the drug is, if it's not going to work at all on your disease, there's no need to give it to anybody, right? So you have to make sure if you are using it, it's going to the right patient population. We're running out of time. We only have just over 5 minutes left, so I'm not going to go through all these slides. But I do want to point a couple of things out, because a lot of focus now is on targeted therapies. And you may hear in the meetings the word chemo-free, all right? If we go back, targeted therapies is not anything new. This was a quote from 1904 when they were talking about using the immune system to fight the cancer itself. And so this is a long-term philosophy. They've just found a way to make work now, all right? The other thing to know is when we're talking chemo-free does not mean it doesn't have side effects, right? Some of them, even with these new therapies, the side effects are quite severe and can be limiting for patients. We have patients, a number of patients, that have to stop using therapies not because they're no longer fighting their CLL, but because they cannot tolerate the side effects, okay? So just so we're clear, chemo-free just means it's different, and it works in a different way. It does not mean it works for everybody, and it doesn't mean that it does not have side effects, all right? And this is just really quickly, immuno-oncology. It just means we're using your immune system to fight your cancer. And what I want to show you, there's a test on this later. is this is what's happening in a B cell. These are all the different processes that are at work in any one B cell. When we are learning more about these cells, it gives then people that are researching and looking into finding new drugs all kinds of different targets, different pathways that they can try and stop happening to hopefully kill the cancer cell and get rid of the cancer in a patient. We didn't know all of this stuff for all of this time that we've been doing research, right? It's like as we learn more and more and more and technology gets better and better and better, we're figuring out more things, and that gives researchers new targets to aim for to see if they can actually stop the cancer from multiplying in a different way. And so if you look at this, it's a very modified version of that slide I showed you a minute ago, but it shows you where some of the new drugs that we're looking at right now are attacking or stopping different parts of processes or aim for different proteins on the surface of the cells to see if they can kill the cells in different ways. All right, so you may hear when somebody's talking about a BTK inhibitor, all you have to know, and you're like, what does BTK stand for? It doesn't really matter in the big scheme of things. All you'll know is that it's a different part of that B cell that they're trying to stop a process happening so we can kill the cancer cell. All right, and so what we will hear quite a bit of at this meeting is discussions around definitely BTK inhibitors, like ibrutinib is a BTK. We'll hear about BCL2 inhibitors, which venetoclax is a type of BCL2. There's combination therapies using traditional what would be rituximab that are looking at a surface protein that it attaches to called CD20. So when we talk about combinations and say we're going to use VR, for instance, it's because we're going to attack this pathway and the protein at the top to see if we can kill the cancer cell with more than one thing happening, as Brian was talking about a minute ago. All right? So when the doctors are speaking specifically, don't get too caught up if you don't understand all of the acronyms they are using. Go for the big picture. We do our best to ask the doctors to simplify and remember that they're talking to patient advocates and not physicians necessarily. And we may be very well educated, but we won't necessarily know all of these things. If you get really confused, and write a note. You can come ask Brian, you can come ask me. Michael, you can ask him and he may send you to me or Brian.
I may ask you a question back.
We will do our best to help you. Our doctors are also really good. So if you have questions, go and ask them after. Most of them, I can't think of one who would not be open to talking to you after the fact and helping you understand something. Okay? Because what's really important is you have really good information as you go back to your home organization to help your patients. Okay? And we have lots more slides, but we don't have time for them. So keep in mind, there's going to be certain topics that are discussed at this during the conference because there's still really big issues in CLL. You've heard us talk about testing. It's definitely an ongoing issue for us. We know that with these new therapies that are being developed, they're not for some people, they're looking like they may be curative, not so much for others. So there's going to be discussions on that and how we might want to combine them in the future. What has also happened is there have been so many new therapies that have come out in the last few years that the trials haven't been necessarily comparator trials. So we haven't compared venetoclax to ibrutinib. Right? So that means there's still questions about which drug to use when. So we're going to have doctors talking about that, like where do each of these medications fit in the treatment paradigm? Because right now a lot of it is opinion-based. And so there's opinions that are coming from the German study group and there's opinions coming from different clinics in the US. And so it's like the consensus is still being built. Okay, so there'll be discussions about that.
Well, I make a point actually about
Yes.
that, and we spoke about it last night, and it is only an opinion, but I sat in at one of the sessions at EHA, Dr. Annan Carter, and it was working out which treatment regime would be the best for patients, and where we all might be kind of fighting to get like one drug approved in a country. One doctor's opinion was it may be giving ibrutinib for 3 months, giving venetoclax for 9 months, and then obinutuzumab for another 6 months. And he had that may be a cure. So this
Yes.
is, and all different opinions are coming from the doctor. So it's
So many opinions. Yes. So many opinions about what we're going to do with this. And I will tell you where we struggle as patient advocates to keep up with all this knowledge and what has come out in the last, few years. Doctors are also struggling. Brian referred to the fact that, some doctors just can see test results and still not know what is the best treatment option for patients. So there is a struggle to keep our physicians up to date as well. When we'll talk about later why we're meeting with IWCLL, and it's a very elite group of doctors that are usually at IWCLL. For your average community oncologist who's trying to keep track of not just CLL but other cancers and how to best treat, this is really hard for them to stay on top of, especially if treatment guidelines haven't been updated and they don't have like a little formula they can follow. So there's still lots of work on that, and you'll hear some of that discussion as well during this meeting, and that's why. Okay, so in like the last minute, any famous last questions?